by coincidence sort of fits in where I end up in this order here because Professor Hall talked about DNA getting the protein, and then Professor Harris talked about looking at the protein and the function of the protein. And she said one of the reasons you do that is fit into the active site and control the protein. Well, I make molecules. Do synthetic work with the purpose of looking at how they interact with biological systems. We don't do much with biological work. We mainly make the molecules, but the whole first part of my talk is talk about why we make the molecules. So we're interested in the enzymes that process carbohydrates. They're the molecules, the biological molecules we're aiming for. And so carbohydrates are sugars or saccharides, oligosaccharides, polysaccharides. As far as biological systems, they have lots of function. One of which is their chief energy source. They also are gross structural support. Wood is mainly made up of carbohydrates, cellulose. The exoskeleton of insects, the chitin or of crustacea, is mainly carbohydrate. So very important structurally. They are the structural backbone for RNA and DNA. Deoxyribose and ribose are sugars, they're carbohydrates. Where even more interesting, though, is cellular recognition. The surface of cells have carbohydrates on them that make them the types of cells they are. They make the immune system recognize them as cells or not cells. Also, it's very important for cellular adhesion. So cells stay hooked to where they're supposed to be and hooked to each other by their carbohydrates. If a cancer is going to metastasize, okay, there has to be changes in its carbohydrate surface in order to break away and go through the bloodstream. So these carbohydrates are very important for that. Also, viral recognition. There are many viruses that recognize the cells are going to infect because they have proteins on the surface of the virus that recognize the carbohydrates on the surface of the cell, and then they know that's the cell they're supposed to go after. I want to talk about the simplest carbohydrate is actually a monosaccharide. And this is an example of glucose, probably the most common monosaccharide people know of from an energy standpoint. And notice this very simple 6-atom ring. For those people who have had organic chemistry, there are 5 stereocenters in glucose. Okay? That means there are 32 different stereoisomers, okay, for this one molecule. They're not all glucose. This is glucose here. You can change the stereochemistry here and we still call it glucose. But these molecules, these carbohydrates, have a lot of variability because of all this stereochemistry. And all that variability is why they're so important for recognition, because you have these small changes. Often, for those who have organic chemistry, know we often draw our structures without drawing out the carbons and hydrogens on carbon to make it easier. So this is how we normally draw our structures. But in reality, we need to be thinking when we see these structures that they have 3-dimensional shape and they fill space. Okay? This is a more accurate representation of what this molecule would be. Okay? It has a 3-dimensional shape to it, and that shape is what's going to be important because of that stereochemistry. So, again, familiar with cell recognition. Blood type. People know about A blood type, B blood type, O blood type. What that means is your red blood cells have different carbohydrates on the surface. If you have O, you have this carbohydrate. If you have B, you have that carbohydrate on the surface of your red blood cell. If you have A blood, you have this carbohydrate on the surface. If you have AB, you have both of those. And you know if you have the wrong blood type, your immune system, you get the wrong blood, your immune system says, wait a minute, that's the wrong carbohydrate, we need to kill those off. And you have an immune response to the blood that you have. HIV. HIV on the surface of the cell has a protein that has a binding site for a carbohydrate. This carbohydrate happens to be the carbohydrate on the surface of the CD4 cells. So the way they find the CD4 cells of the immune system is by this carbohydrate that's on the surface, and oh, that's the cell I need to infect. So, think about this, if you can make a molecule that looks like this, that binds into this pocket, it would bind into the HIV binding site, blocking it, preventing it from binding to the surface of the CD4 cells. So this is why we're interested in making these types of molecules that block carbohydrates. So for a simple example of where this comes into play again is the idea of energy, food. Okay? This is a piece of bread. We can eat bread, has starch in it. We have enzymes in our digestive tract that will break down the starch into free glucose molecules. Chunk of wood, made of cellulose. You know, most of us don't eat wood unless we eat shredded wheat. At least I think it tastes like wood. I don't know. But the molecules here, it's still glucose. The difference is stereochemistry right here. They're glucose molecules, stereochemistry is different, shape of the molecule is different. We have enzymes that recognize this shape, that digest starch. Termites, actually bacteria in the gut of the termites, have enzymes that recognize this shape to hydrolyze that into glucose. So, talked about enzymes. My movie is actually a very simple cartoon movie about how these enzymes would work. So again, because we're going to make molecules that inhibit these. But the idea is the substrates come in, the small molecules, they bind to this active site on the enzyme, and that's where the chemistry takes place. Okay, that's the catalyst takes place, and then they're released. And if we make a molecule that will bind in here real tightly, we can block that active site, prevent the enzyme from doing what it's supposed to do. So, what do we try to do? How do we do this? So we're interested in, this is our glucose again. We make amino sugars, remember the title. Amino sugars are compounds that look like carbohydrates, look like the sugar, but they have nitrogen instead of oxygen. Okay? So we have these amino sugars. These look like the product of that hydrolyzing cellulose, or hydrolyzing starch. The reaction along the way, when you look at the reaction real closely, this is an intermediate. This cation is an intermediate of that reaction with hydrolysis. These inhibitors, these molecules look like the intermediate. Often they end up being better inhibitors. They bind more tightly. So we're interested in making these molecules that look like the sugars, have a nitrogen in place of the oxygen, because they have a tendency to bind the enzyme tighter, and they're not processed. They bind in there and they stick. So our goal is to make these, again, remember there are 32 isomers of this. So we want to make compounds of this type or this type that have all different stereochemistry. So if this molecule here has 3 different stereocenters, it's 8 isomers. So it's not quite as bad as glucose. So what we did is we designed a synthesis that we could choose a sugar to start from, do a series of steps, and get out the 8 different isomers of this pyrrolidine. And this is what we do, the synthesis. So the pyrrolidine synthesis, here's your synthetic scheme that we've come up with. Okay, we're going to talk about every reaction in detail. Right? No, okay. The idea is, what we do is we do this chemistry, the product of this one we have to use in the next reaction, and we have yields here. We like all our yields to be better than 60%. Okay, because the idea is that it carries to the next step. And so we worked on the synthesis. Here I started with galactose as the sugar. I changed the stereochemistry here. We can start with any of those 8 sugars and get any of the different 8 ones here. This work was done by, you know, this Brammer work that, you know, people that work for Hall. It's the same Leanne Brammer. Same Leanne. She actually did research with me before she spent the summer doing research with Professor Hall. And some of this work is hers, and the last stuff here is Sarah Rogers. Notice this step here, kind of low. We still need to work on that. That's one of the things we're working on. Another thing I want to notice is this step right here is a key for all the types of molecules we make. We change the amine, we get a different product here, we can make different types of compounds. So, indolizidine, same compound, remember there? We change the amine, we get the diene here. Another key reaction in a lot of synthesis we do is this metathesis, where it takes 2 double bonds and joins them together to make a ring. So we can take this molecule that has 2 double bonds, add the ring, and we have this indolizidine. And this synthesis was actually finally finished this summer by Kirsten McAndrews. It was started by Samantha Sweeney, and some of the original stuff to get to here was Ellen Sledge. And the last sort of synthesis is pyrrolizidine work. This now used mannose. Difference I want to point out here is all the other ones didn't have these 2 methyl groups. It turns out this is something that had to be worked on. When you have a normal benzene ring here, the yields here are about 40%. But when we add the methyl groups, we get a much better yield. Types of things that work. So what do we do? My last slide. I know I used up my time. We plan the potential synthesis. We do a lot of literature reading, see what people have done before to improve these steps we're trying to do. We then have to get the reagents that we need to do the synthesis. If the reagents aren't as pure as we want, we have to purify them. Big thing we do is we run the reactions. Once we run the reaction, we end up with having to separate the product from the reagents and the byproducts and stuff, and we often have a mixture of products. And then we have to purify each product, and then we have to go through by running spectroscopy and identify what they are, figure out everything we had, decide if the reaction works satisfactory, maybe give us nothing we want, maybe give us a little bit of what we want, and whatever, and then either go on to the next reaction if it works satisfactory, or go back and try again. We do a lot of going back and trying again. That's why they call it research. You keep researching it over and over and over until you get things that work the way you want. Thanks.